Import and sorting of chloroplast proteins. Although chloroplasts encode more of their own proteins than mitochondria, about 90% of chloroplast proteins are still encoded by nuclear genes. As with mitochondria, these proteins are synthesized on cytosolic ribosomes and then imported into chloroplasts as complete polypeptide chains. They must then be sorted to the appropriate location within chloroplasts, an even more complicated task than protein sorting in mitochondria, since chloroplasts contain 3 separate membranes that divide them into 3 distinct internal compartments. Protein import into chloroplasts generally resembles mitochondrial protein import. Proteins are targeted for import into chloroplasts by N-terminal sequences of 30 to 100 amino acids called transit peptides, which direct protein translocation across the 2 membranes of the chloroplast envelope and are then removed by proteolytic cleavage. The transit peptides are recognized by the translocation complex of the chloroplast outer membrane called the TOC complex, and proteins are transported through this complex across the membrane. They are then transferred to the translocation complex of the inner membrane called the TIC complex and transported across the inner membrane to the stroma. As in mitochondria, molecular chaperones on both the cytosolic and stromal sides of the envelope are required for protein import, which requires energy in the form of ATP. In contrast to the pre-sequences of mitochondrial import, however, transit peptides are not positively charged and the translocation of polypeptide chains into chloroplasts does not require an electric potential across the membrane. Proteins incorporated into the thylakoid lumen are transported to their destination in 2 steps. They are first imported into the stroma as already described and are then targeted for translocation across the thylakoid membrane by a second hydrophobic signal sequence which is exposed following cleavage of the transit peptide. The hydrophobic signal sequence directs translocation of the polypeptide across the thylakoid membrane and is finally removed by second proteolytic cleavage within the lumen. The pathways of protein sorting to the other 4 compartments of chloroplasts, the inner and outer membranes, thylakoid membrane and intermembrane space, are less well established. As with mitochondria, proteins appear to be inserted directly into the outer membrane of the chloroplast envelope. In contrast, proteins destined for either the thylakoid membrane or the inner membrane of the chloroplast envelope are initially targeted for import into the stroma by N-terminal transit peptides. Following cleavage of the transit peptides, these proteins are then targeted for insertion into the appropriate membrane by other sequences which are not yet well characterized. Finally, neither the sequences that target proteins to the intermembrane space nor the pathways by which they travel to that destination have been identified. Function of chloroplasts. Photosynthesis. During photosynthesis, energy from sunlight is harvested and used to drive the synthesis of glucose from carbon dioxide and water. By converting the energy of sunlight to a usable form of potential chemical energy, photosynthesis is the ultimate source of metabolic energy for all biological systems. Photosynthesis takes place in 2 distinct stages. In the light reactions, energy from sunlight drives the synthesis of ATP and NADPH coupled to the formation of oxygen from water. In the dark reactions, so named because they do not require sunlight, the ATP and NADPH produced by the light reactions drive glucose synthesis. In eukaryotic cells, both light and dark reactions of photosynthesis occur within chloroplasts. Electron flow through photosystem 1 and 2. Sunlight is absorbed by photosynthetic pigments, the most abundant of which in plants are the chlorophylls. Absorption of light excites an electron to a higher energy state, thus converting the energy of sunlight to potential chemical energy. The photosynthetic pigments are organized into photocenters in the thylakoid membrane, each of which contains hundreds of pigment molecules. The many pigment molecules in each photocenter act as antennae to absorb light and transfer the energy of the excited electrons to a chlorophyll molecule that serves as a reaction center. The reaction center chlorophyll then transfers its high energy electron to an acceptor molecule in an electron transport chain. High energy electrons are then transferred through a series of membrane carriers coupled to the synthesis of ATP and NADPH. The proteins involved in the light reactions of photosynthesis in plants are organized into 5 complexes in the thylakoid membrane as depicted in the image. 2 of these complexes are photosystems 1 and 2, in which light is absorbed and transferred to reaction center chlorophylls. High energy electrons are then transferred through a series of carriers in both photosystems and in a third protein complex, the cytochrome BF complex. As in mitochondria, these electron transfers are coupled to the transfer of protons into the thylakoid lumen, thereby establishing a proton gradient across the thylakoid membrane. The energy stored in this proton gradient is then harvested by a fourth protein complex in the thylakoid membrane, ATP synthase, which, just like the mitochondrial enzyme, couples proton flow back across the membrane to the synthesis of ATP. One important difference between electron transport in chloroplast and that in mitochondria is that the energy derived from sunlight during photosynthesis is not only converted to ATP but also used to generate the NADPH required for subsequent conversion of carbon dioxide to carbohydrates. This is accomplished by the use of 2 different photosystems in the light reactions of photosynthesis, one to generate ATP and the other to generate NADPH. Electrons are transferred sequentially between the 2 photosystems, with photosystem 1 acting to generate NADPH and photosystem 2 acting to generate ATP. The pathway of electron flow starts at photosystem 2, which is homologous to the photosynthetic reaction center of R virid is already described. However, at photosystem 2, the energy derived from absorption of photons is used to split water molecules to molecular oxygen and protons. This reaction takes place within the thylakoid lumen, so the release of protons from water establishes a proton gradient across the thylakoid membrane. The high energy electrons derived from this process are transferred through a series of carriers to plastoquinone, a lipid soluble carrier similar to coenzyme Q of mitochondria. Plastoquinone carries electrons from photosystem 2 to the cytochrome BF complex, within which electrons are transferred to plastocyanin and additional protons are pumped into the thylakoid lumen. Electron transport through photosystem 2 is thus coupled to the establishment of a proton gradient which drives the chemiosmotic synthesis of ATP. From photosystem 2, electrons are carried by plastocyanin, which is a peripheral membrane protein, to photosystem 1, where the absorption of additional photons again generates high energy electrons. Photosystem 1, however, does not act as a proton pump. Instead, it uses these high energy electrons to reduce NADP plus to NADPH. The reaction center chlorophyll of photosystem 1 transfers its excited electrons through a series of carriers to ferredoxin, a small protein on the stromal side of the thylakoid membrane. The enzyme NADP reductase then transfers electrons from ferredoxin to NADP plus, generating NADPH. The passage of electrons through photosystems 1 and 2 thus generates both ATP and NADPH, which are used by the Calvin cycle enzymes in the chloroplast stroma to convert carbon dioxide to carbohydrates.